going to finish up Module 7, and I'm going to send you home with a quiz, a take-home quiz, which means you can, yes, use your notes to take that quiz, but you must bring it back to me next week. If you don't bring it back, I can't give you credit for it.
So are you telling us we can use our notes, or just accepting that we will? So
You can. I'm giving you permission to use your notes. No.
Yeah.
I didn't say your book, I said your notes. That means you have to take really good notes. Okay? Also, you do need to actually read through the module, of course. We'll have our midterm next week, so when you come in, you're gonna come in, sit down. I'll hand you an answer sheet, a midterm exam. You will take the exam. I have the tables arranged differently, so we're not so close to one another. It is a multiple choice exam, and then once you finish it, you're free to go. Okay, and so then I'll be grading them. Right then, if you want to hang around, you can probably come back in a few minutes and see if I've got them graded. I need you to bring those lab books. That's very important. I'm going to give you a lab grade next week, and the lab grade is the single thing that can bring your grade up really quickly. I also will drop the lowest quiz grade, okay? 1 per year, not per semester. Okay, lowest quiz grade will be gone. So if you have a really lousy quiz grade, Jupyter will do it automatically. I'll go in there and tell it to drop the lowest quiz grade. It'll drop that. You're taking tests at home. You need to record those in the back of that book right there. I need them all in there, and I'll put all of those into Jupyter. And therefore, after next week, I'll look at that. I'll put them all in, and then after next week, you'll be able to see your very accurate grade for right now. Right now, looking at just your quiz grades is not an accurate reflection of your What are you bribing me? Oh, this is tuition? Yes. I'll take it. I'll take a bribe too. No, I'm kidding.
Tab on your desk is an early Christmas present, but I will give you one.
Is it from you?
Yes.
This is?
Yes.
Oh, thank you. Thank you.
Somebody gave it to me.
Lovely.
You gonna give me my candy boy?
Oh, it's a regift?
Yeah. What?
Okay, here we go. So, also next time, I don't think we need calculators for this class, right? I'm just thinking about, bring your lab book, bring something to write with. I don't think you need to bring anything extra for this class. Okay, we're going to finish up Module 7, working with genetics. Who's that man right there we're looking at on the screen? Gregor Mendel, very good. Remember, he's the one who worked with the English P's, and because of his work, we figured a lot of things out. We're going to look today at sometimes genes don't follow Mendel's rules, okay? So even though Mendel figured out the general idea of genetics, we call that Mendelian genetics, there are some things that don't fit in, and we call those non-Mendelian traits or circumstances. All right, so here we go. I want everybody to take your hands like this, okay, and then fold them together like you're going to pray with your fingers interlocked. Okay, show me what thumb is on top. Right, left, right, left.
I could just go
Left,
like this.
left.
No, just do
Right.
this.
Left.
We're just
Okay, now take your hands.
Okay, Miss Gasper, is this
I
correct?
need it next week.
Is this correct?
Yes.
Okay, next what I want you to do is take them and rearrange them so the other one's on top. The other thumb is on top. And feel how strange that is. Okay, it's very odd, isn't it? Okay, so it turns out that whatever you do this way is genetic. All right, this is the same thing. Have you ever noticed, for instance, I know if you're right-handed or left-handed, that's a big one, right? What about listening on the phone? When you put the phone to your ear, what ear do you put it to?
I don't put it both.
Some people
No,
left, some
speaker.
people
I
right.
don't put it. I do both.
I don't put it to my ear. I do like this.
Oh, you
I'll probably use
What are you,
my
60-year-old
hand,
man?
switch it if
So
I need to
I have a
use
friend.
Quiet.
I have a friend who was born right-handed.
Quiet.
I have a friend who was born right-handed, and when she was like 6, she accidentally put her hand on the stove and it completely burned her hand. And so she recovered like within like a month, but she completely went back to, she was left-handed from then on.
Okay, yeah.
And she's never swapped back.
Yeah, we can practice. I started off
Well,
kind of
I think when she gets her
ambidextrous.
right hand back, she could be like ambidextrous.
I'm a little ambidextrous, but it was very much easier to write right-handed because of the smearing situation.
I'm left-handed, and this is my priority hand. But for anything other than writing, like if I'm like baseball, when I'm swinging, when I'm pitching, when I'm throwing, anything, I throw with my right.
Interesting.
I swing
I
with my
will
right,
tell
everything
you,
is right.
while putting your hands this way is genetic, left-handed and right-handedness is not. In fact, it's still somewhat mysterious as to why we have left-handed, right-handed. I will tell you, in the whole animal world, the only animal that shows a preference for handedness is humans, and that is a preference for
What about
We're not
right-handedness.
animals.
That's because most other animals.
For right-handedness,
What
okay?
about-
what about
And
monkeys?
About
Oh, mammals, they're
87% of the population is
man.
right-handed.
See, look, what we
Question,
call- what we
question.
Which one is more common?
So it's interesting, those of you who had your left thumb on top, but you're right-handed, that's why I asked about, what if when you're shooting a gun, do you aim it with your right hand or your eye or your left eye?
Right.
Right.
What about you?
So here, again, this is a weird one.
See,
Trigger
I
with-
just use
My
a
left eye is dominant, but I always aim with my right eye.
And you pull trigger with your right hand. Okay.
It's
If you're right-handed and your left eye, you have to kind of sort of lean your head way over.
Oh, I have
My
a friend
husband's
that does
that.
that.
Yeah. Whereas this is a little more,
I have a
yeah.
friend that's like right-handed, but he shoots with the left, and he's way better at it.
Okay, okay.
I see.
All
I
right.
just punch him in the nude codes.
Now the reason, y'all were supposed to go home and look at your family's earlobes.
Oh, I didn't do
I
that.
did. I did. I had the
And
same
what's
one.
nice about the earlobe situation, it's on a single gene. Okay, we're gonna talk in a little more depth today about eye color. It's more complex than I even realized. I told you it was very complex. They're figuring out now it's even more complex. So we're gonna see that the dominant earlobe is the unattached like this versus attached like that. And so if you're looking closely, you can see how it's attached on B here and not here. Typically, you can tell easily
Mine
if an
is in
earlobe is
between.
attached.
Mine's
Okay.
attached,
So
I'm
y'all were
humble.
supposed to.
And humble. Very
Y'all were
humble.
supposed to do a pedigree. This is my pedigree of my family. My husband has attached earlobes. My earlobes are detached. I have 5 children, 1 daughter and 4 sons, and they all have detached earlobes.
You
So
only
you
have-
can see
you have 4 sons?
clearly, I have 4 sons. You see clearly that the detached is the dominant trait on this particular trait. Yes.
Is pedigree a dog food?
Pedigree is also, you say, like if you're looking at the lineage of a dog, you know, like say a purebred dog. That's why they named the dog for that. Because pedigree talks about, like if you have a purebred dog, it's referring to its pedigree.
Oh.
And that's this right here, to say that it came from German Shepherd to German Shepherd, it's all German Shepherd, so you know that they're officially a German Shepherd, that kind of thing. All right, so I'd like you to still do that. You are also supposed to do your dihybrid cross between the, this. You were supposed to do this with the flowers. Did y'all end up doing that in your book? And does it look something like that?
I did.
Yes,
I did
but
one
I
for
was
the study
confused.
modules.
What,
Yeah, I believe the answer was right there in your textbook.
I thought ours
Oh,
was
no,
like
it wasn't.
FT. Wasn't ours with like FT?
Was it FT or Ps?
Yeah, because you showed us on the screen.
Oh, our previous one for the previous
I filled it out.
edition of the
I have
book was
the
P.
percentages.
Okay, is it filled out in your lab book?
Yes, and
That's
I have
good.
the same percentages.
Remember, I'm gonna grade your lab book next time, so if you don't have it filled out in your lab book, then get that done by next week, so that you can have that good grade for that last lab. Alright, so let's look at genetics and inheritance. How many genes do humans have?
45.
I'm sorry, I take that back. How many chromosomes do humans have?
23.
23 pair,
46.
which is 46,
I have
right?
25.
Don't you have 22?
Okay, here we go. Our first set of blanks. Humans have 23 homologous chromosome pairs. Remember, homologous means they have the same genes on them. One from your mom, one from your dad. So one chromosome from your mom has genes X, Y through Z, whatever it is, and its homologous pair has your father's for those same gene choices. Pairs 1 through 22, guys. Pairs 1 through 22 are called the autosomes. The autosomes. And I'll show you here something called a karyotype. Here we have 1, 2, 3, all the way down to 22. And notice in 23 is a special group. So 1 through 22 are called autosomes, and 23 are called the sex chromosomes. Okay, so like we have 1, 2, 3. Notice they're kind of a little different looking from one another. And look how like number 13, 14, and 15 have kind of a truncated top right there. But they're matching each other, and on those you have some genes that are coding for certain things. And we like to say that we have the entire human genome coded. We know where they are and what chromosome they are. But after the whole study I did on the eye color, we're still finding out so much stuff about the chromosomes and the genes that affect other genes and this kind of thing. Okay, so let's take a look at the sex chromosomes. So if we're going to do a Punnett square, let's say a lady gets pregnant and she'd like to know, am I having a boy or a girl? Well, we do a Punnett square about them. What we know for a female, she has 2 chromosomes to add. one each, right? So you can send one this way and one that way, and they're both X. The male has an X and a Y. Notice the Y is cut off kind of short like that. That's going to become important. What?
Why do the X's not look like X's?
Well, because that's not why they're called X's. But I remember how we look at chromosomes when they are X-shaped. It's because they've been duplicated.
Right.
These have not been duplicated.
Oh.
Okay. All right. So if we have the female right here, she's XX. We have the male who's XY. We do our Punnett square. We'll see every time we're going to have a 50-50 chance of male and female. So anytime a woman gets pregnant, she has a 50-50 chance of having a girl and a 50-50 chance of having a boy. Okay, every time.
That's why it's mostly 50-50 in the entire world.
It's kind of 50-50. I know that recently we've seen that the female population has been like 50.4 or something
Nobody
like
cares
that. There's
about
a
men
few.
anymore.
I thought it was
Yeah.
like literally 50.
And then there's questions about the DNA being affected by certain things like drugs. And what I mean by drugs are
drugs.
things
Drugs.
pharmaceuticals would be one, but things like we treat animals with certain antibiotics that they keep them from getting sick so they can stay
Right.
alive long enough to make steaks for us. So we get these toxins in our food, and sometimes those toxins may affect DNA. We don't know yet, but it's possible. And if that's the case, some people have even said the Y gene is disappearing, and that, of course, would be the end of
What?
mankind.
No.
No.
Why?
I don't know. Do you have a spot to write down?
I like, it says calculate the percentages of traits
Oh,
of
yeah.
male and female. So I just like wrote it down, so like.
Humans are done.
It says the Y. Yeah. I think that first box in your book is talking about fruit flies. So let's wait till we fill that in. This is a real simple Punnett square. You may be asked on your, you would definitely be asked on this quiz, but probably on your midterm to do some Punnett squares. So I want you to feel comfortable doing the Punnett squares. I don't typically have the dual trait Punnett square, in other words, the 16 square Punnett square, just the 4 square Punnett square.
4 square.
Okay, pair 23 is called the sex chromosome pair. That's the blank you're filling in there. We use X for female, Y for male. Interestingly, the Y chromosome does not carry any traits. And it's very short. It's missing a part of it right there. So if there's a gene on this part of the X chromosome that's where the Y is not, then that can be expressed sometimes because there's nothing on the Y to counteract. In other words, there'll be only one gene for that trait. Sex-linked genetic traits are traits that are carried on the chromosome pair 23 on the X chromosome. Okay, so there are some genes on the X chromosome, and there's nothing on the Y to match up against it. Okay, there's no homologous pair for that gene. Do you get it? So there'll just be the one gene. That's it. Okay, so it turns out that some of these have become quite famous, actually. All right, so the sex-linked genetic trait exists because the Y chromosome doesn't carry as many, it says here as many alleles as the X. That sounds more accurate to me. These traits have an allele on the X chromosome and not on the Y, so the male will end up expressing that. Okay, so when that happens, so if there's a gene, like right up here, there's nothing to counteract it on the Y, then that gene will be expressed in the male. In the female, that gene has a second gene to influence it, whether it's recessive or dominant, right? So we know about recession and dominance. So let's look at this. This makes it more clear. We can see the gene is up here on the top of the X chromosome. The Y has nothing to cover it up. That's the way I like to say it. Has nothing to counterbalance it, to cover it up. Therefore, whatever that gene is, it will be expressed in the male. And this happens to be the gene for color blindness.
So that's why most colorblind cases are guys.
That it's an X-linked gene. So therefore, we could technically say that almost all color blindness is found in males.
That's not fair.
There is a way for a female to be colorblind, of course, but her father has to be colorblind in order for that to take place. So everybody take a look and tell me, please, that you see something here.
74.
21.
You say 71?
31.
Colorblind.
No.
Colorblind.
Do you see, if you're colorblind, you don't even see a difference between anything. You see a big red blob, and that's all you see.
Yeah, I'm not color.
Everybody can see it?
Yeah.
I'm looking at you guys.
Yeah, so
Oliver, can you see it? I know.
What are you, Lord?
All right. So that's good news. Cole's listening at home. Cole, can you see a number 74 in there? Okay, hopefully you can. If you cannot, you might have color blindness, or I have a lousy test right there. All right, so that's an X-linked gene.
No,
so,
leave it off, leave it off.
why are we doing that?
No, I just mean leave the lights off.
Turn the lights
Oh,
off.
lights. You want the lights off?
Yes.
Yeah.
Okay. All right.
Oh.
So the reason we figured this out, y'all, about this X-linked gene, we probably would have figured it out sooner or later, but there was a guy doing studies, and he realized that studying humans, okay, remember Mendel was studying the peas, and every time he had to figure out the trait on the pea plant, he had to grow a whole new set of peas and wait for them to mature. That takes a lot of time. Furthermore, there are lots of genes on the peas. Remember all those factors he was following, all those genes? We had like 8 of them, and I think there were even more of them. And so he was kind of not losing track. We were starting to lose track of those. And so this one guy comes along and he says, I wonder if we could study something that was simpler in its genetics. And it turned out he picked the fruit fly. The fruit fly has 8 chromosomes. 8 genes. I think it's 4 chromosomes and 8 genes. It's really, really simple on the fruit fly. Yes.
On the 2 diagrams, why does it show like whenever the mutant? Why does it say short antenna and then for the rest of them it just has them long?
Okay, so here's what I'll tell you. When we're looking up here on the top
No,
here,
I mean like on the bottom.
I understand.
It
I have
says
to start here in order to get to that. The type of this is called the wild type on the top here. The vast majority of the fruit flies that were being studied looked like this, and they had red eyes, they had a gray body, they had normal wings, and why are we talking about red eyes again? Apparently the red eyes are coded for on these genes right here. All right, in the mutant version, I'm sorry, this is not red eyes. The first one was long antennae. So the gene here codes for short antennae here. The vast majority of you see them are right here. Now what's nice about fruit flies is you put them together. Now when I was in high school, I actually raised fruit flies. We had it as an experiment. I would love to do it here, but we can't because you have to be here every day to take care of them. We would take the weekends off, of course. But you put them in a little vial with food in there, and they reproduce. So you start with 2, a male and a female, wild type, red eye, and they go in there, and then every now and then you're going to see a mutant type come out. So they called it mutant type because it went against the normal. So we would possibly, this is before they understood the whole, they understood recessive, but they didn't realize how they were being dealt out as recessive, okay? This is the same thing as recessive. All right, and so what the big breakthrough came with, actually it was white eyes. So I'm surprised here. Look, this cinnabar eyes, red, not quite red. Here, red eyes, wild. Brown eyes, it says here, but it was really the white-eyed fly that broke open everything for the genetics world. So here's the guy's name is Thomas Hunt Morgan, and he noticed one day when he was looking at his fruit fly culture. When I did these in high school, we had these little cultures, right? And then you would take this little vial about this big and dump them into a little case that had ether in it, and they would fall asleep. Then you would look at them under the microscope and see all these little traits and put them back in. We didn't kill them. We put them back in and let them wake back up.
Jeez.
The only thing is that sometimes a student might leave the cap off a little bit, and so our whole classroom, of course, was filled with fruit flies all the time. And so I think the school eventually said, that's it with the fruit flies. So here we go. Here's our wild type. All of them, male, female, had red eyes, and then all of a sudden a male with white eyes popped out of there. And that's what, when Mr. Morgan figured out there's an issue happening here of dominance, okay? And so he was able to do a Punnett square and all that kind of stuff. And then he started looking and watching. By the way, these are 2 males. I can tell they're males because they have a dark rear end. Females do not have the dark rear end. So it's easy to tell the difference between male and female. And we used to follow several traits: eye color, antenna size, and something called vestigial wings. So their wings sometimes would come out all shriveled up and unusable. So a fruit fly without any wings is not a good fruit fly. Okay, so here we go. A heterozygous, this is what's going in your first box. Can y'all look?
Yes, sir.
Wait, what?
In your first box, you're going to be writing down a Punnett square for this problem. Ready? A heterozygous red-eyed female Drosophila fruit fly is crossed with a red-eyed male Drosophila fruit fly. Will any of the offspring be white-eyed? What is the percentage chance of getting a white-eyed female? What is the percentage chance of getting a white-eyed male? All right, so you're going to write this down in your book. So we know that the eye color is X linked. It's on the X gene. It was Thomas Hunt Morgan who figured that out. So we're gonna see X with the R for red eye, and Y has nothing, right? No eye color. And then we have is this the female? Yeah, XX. X with the R for red eye, and X with the lowercase r, meaning the white-eyed variety. So we're going to bring those down and follow that kind of that superscript right there. So we're going to have big X, big R, big X, big R. Those R's, by the way, are superscripts. They're set above. It's not like the one we were doing in the 16 square deal. I need you to shush. And so that's a female with what color eyes? Red. All right, over here we're going to see XR, big R with Y. That's a male with what color eyes? Red. We're going to come down here. We're going to have XX, big R, little r. That's a female with what color eyes? Red. And here we have Y and X with a little r. It's a male with what color eyes?
What's the other?
White. White eyes.
So highlighting.
Okay, so because Y has no gene to counteract that recessive gene right here.
So how is the top right one?
It's got the big R for red. That's the only gene it has. So this male, he's got this plus a red-eye gene. The female has a red-eye gene and a red-eye gene, and down here on the bottom has a red-eye gene and a white-eye gene, but the reason she doesn't express a white eye is because she has the dominant R there. Now, is this a problem for the fruit fly? The fruit fly can see perfectly well. He's not blind. He gets along just fine. So this is not an issue, except that he, from our perspective, looks different. I don't know if the other red-eyed flies shun him because he's wide-eyed. I don't know. They didn't seem to notice anything strange like that. And so, but we noticed also when I asked you this problem, you understood what heterozygous meant, right? Heterozygous, one for big R, one little r. Not the same. Okay, so 100% red-eyed females. One is a carrier. She's called a carrier because on this X gene she does have that little gene. She can give that out, but likelihood of her being wide-eyed is not very good. Now the males, we have 50% of the males are wide-eyed. Do you get it? Yes, David.
I didn't know that they used fruit flies in World War II. You know, as carriers and stuff.
Shush, dude, shush.
This was happening in early 1900s.
Oh, okay.
Yeah, early 1900s. What they used, but what they found out from this was
Sorry.
earth-shattering. I don't typically have to think of a carrier. You think a carrier pigeon? Is that what you're thinking of? I don't know how that relates to World War I. Okay, so there is a condition, however, that's X-linked, and it's not beneficial to the.
Isn't that what
Bear
happened
others.
with the royal family a lot?
Yes, we're going to talk about the royal family. All right, that's called hemophilia. Hemophilia. I don't know if you know anybody who's a hemophiliac, but if you do, I will guarantee you he's probably a boy. And it's because it's an X-linked gene.
What is it?
Hemophilia.
What is it?
Oh, what's hemophilia? Hemophilia is a disease that you are missing clotting factors and you're unable to clot your own blood. Okay, we're going to watch a little video on it. Okay, shh, let's listen.
So, what is hemophilia? Hemophilia is a bleeding disorder, a problem with a person's blood that makes them bleed longer than a person without hemophilia. No one can catch hemophilia. It's an inherited condition you have from birth, for your whole life. It's just part of who you are, like the color of your hair or the shape of your nose. Here's how it works. Your blood travels through your body in tubes called arteries, veins, and capillaries. Normally, when there's a tear or hole in one of these, bleeding starts, and different parts of the blood go to work to plug the hole. Blood cells called platelets gather at the opening, and other blood parts called clotting factors help hold the platelets together to form a clot that stops the bleeding. When a person has hemophilia, he doesn't have enough of one of those clotting factors he needs to form a clot, and he keeps bleeding longer than he should. Each person with hemophilia is missing one of two kinds of factors. If you're missing factor 8, you have hemophilia A. If you're missing factor 9, you have hemophilia B. Either one can be mild, moderate, or severe. Without regular treatment, having hemophilia means you'll bleed longer than you should if you get cut. But you don't have to have a cut to have a bleed. Sometimes, especially when you're active, the tiny capillaries inside a person's body will bleed, causing a bruise or pain in the joints. Not dealing with those kinds of bleeds can cause a lot of problems later, including arthritis at a young age. But the good news is, there are good treatments available that can help you manage hemophilia effectively. Treatments that replace the missing factor help your blood clot more like a person who doesn't have hemophilia. These treatments, when used properly, help you keep your bleeding under control so you can live a long, healthy life and follow your dreams without hemophilia ever holding you back. To learn more about hemophilia, visit worldhemophiliaday.org.
Diseases these days are also sexist, so everybody's in the woman's favor.
No, these are just X-linked genes. Yeah, these are X-linked, and this happens to be one that's kind of famous, okay?
Exactly.
So the deal is, if a boy ends up with hemophilia, is it possible for a female to have hemophilia? Sure, but her dad has to be a hemophiliac for her to get it. And her mom has to be a carrier.
She exists.
Well, so what we have now is we have medications that have the clotting factors artificially that we can use to give to these people, okay? Now, I will tell you this, that looking the way blood clots, it goes through these factors, and they have those Roman numerals to them, and we have factors coming from intrinsic and extrinsic pathways down to factor X or factor 10, and then coming down from there. And it's really an amazing cascade of biochemical events that takes place. It's like a waterfall, and if you miss one of them, that you will not have your blood clot. And if your blood doesn't clot when it needs to clot, you will die. So there have been people who have studied the clotting of the blood and have become believers in God, because it's absolutely impossible for a step to take 1,000,000 years to take place, otherwise the organism would have died. All right, so it's a pretty fascinating deal. But this hemophilia kind of pops out, and people didn't know why until the 1900s, when we started seeing these X-linked genes. So the most famous example, of course, is the royal family of England. This is Queen Victoria standing right here. Her husband Albert is in the middle, right there. And what Queen Victoria did not know was that she was a carrier of hemophilia. And because she was a carrier, she would then be able to extend that trait to a child. But since her daughters would have two X genes, who would she extend that trait to? Her sons. All right, her husband was also her cousin, and so there is something to be said when you don't have a wide gene pool, that sometimes those genes might become a little more concentrated, especially if the children also marry cousins. Oliver.
Was that kind of the same case with like Tsar Alexander?
Yes, that's one of the children.
They were?
That's
Yeah.
Tsar Alexei is Queen Victoria's grandson.
Oh yeah.
Yeah.
Where's
Actually,
Queen Elizabeth? She in this picture.
not Elizabeth, Victoria.
Yeah.
Not Elizabeth.
wasn't alive.
Okay, so they had 9 children. Let's take a look at them here. Here's their 9 children. Their first child was a daughter named Victoria. She's fine. And then comes Albert. Boy, he's okay. He's fine. Then Alice. I think they go this way. Then Alfred this way. So it turns out, after some time, they did realize that poor little Leopold right here did have the trait. He did have the hemophilia trait. And so when he was very young, here he is, they noticed pretty quickly that he had bleeding incidences that wouldn't stop, and they, of course, had the best medical care in the world. They were starting to figure out that after a few times that it was hemophilia. They knew about the disease. They didn't know how he got it. They didn't realize it was an X-linked gene at this point. And so Victoria never did take responsibility, if you will. Not you can. She had no control over the gene that she had either, but she never really accepted the fact that it was her that gave the gene to her son.
Is it King Alfred?
Yes.
My mom's told me there was this guy that had hemophilia that died of a nosebleed.
Sure. If they don't have the proper intervention, the nosebleed will kill them. The husband's name, whoever asked that, the husband's name is Albert. He's called Prince Albert. He was not a king.
No, it says that Alfred.
Which one?
Alfred Edward.
This one? Oh yeah, Albert. Albert.
The one
He's
below.
Birdie.
One below. His name's Alfred.
Alfred. Okay, right there. Yeah, he's okay. But the only boy that got it was Leopold. So out of 9 children, that's not too bad, right? It would have been a half and half. It would have been 50-50, but they only had one child that actually ended up with it. But their daughter, Alice, was a carrier, and their daughter, who is that? Beatrice was also a carrier. Alice's daughter married the Tsar of Russia, and their son, they only had one son. They had lots of daughters, like 4 daughters and 1 son, and the one son, Alexei, did have hemophilia.
And then they all were murdered
I
by
would say it
cops.
would kill him, except it was the bullet wounds that killed him, probably more than the bleeding disorder. That would have been, they were all killed by bullets. I don't know if you know that. When we get to it in history, it's pretty awesome.
By the who?
It's pretty
By the
Bullets.
communists.
By the Russians.
Communists.
Okay, so when poor little Leopold was growing up and they found out that he had this problem, they never let him go play. So he spent the majority of his life looking out the window watching his siblings play. It was very kind of sad for him. But he did survive into his 20s. In fact, he had 2 children. He got married and had a couple of children. And his son did have hemophilia. I'm sorry, his daughter became a carrier for hemophilia. I think his son was okay. But at about age 23, I think he fell and hit his head and he caused a bleed in his brain and he did not recover from that and he was killed. He died from his hemophilia at that point. All right, so here we go. We have the genotype of the non-hemophiliac husband, that would be Albert, and the heterozygous queen who's carrying. So we have our H for hemophilia, little h right there, okay? And we have our big HY here. So we're going to see if we bring our Punnett square down. We have a female who is fine. We have a male who is fine. We have a female who is a carrier, but she does not exhibit the trait. And then we have a male who is does suffer from the disease of hemophilia. So that's what your Punnett square is going to look like for these guys right here.
Wait, the top left, she would have it.
No, big H means no hemophilia. The lowercase h is what represents hemophilia.
Oh, okay, okay.
The other H would be normal.
Oh.
Yes.
Can I go use the restroom?
Yes. Okay, so like I said, it would have been 50% of the males, but 25% of the children. So they, having just one son that way, they actually made out pretty well. We're going to see right there, that is the guy that's going to have, that's Leopold, poor Leopold. So how do we get a hemophiliac female? All right, so if I have a hemophiliac dad, and he marries a cousin who carries the gene right there, we're going to see right here in the lower left box, do you see X little h, that is a hemophiliac female. It's not that common, but it can happen. So X-linked genes can be passed to females, but the father must possess the trait. Here, he's got it, he's got hemophilia. So today, let's just say somebody who's suffering from hemophilia goes to a hemophilia support group or something, you know, might meet a girl who came from a hemophiliac family, who's got a brother or something like that, and she might be a carrier. So they might, before they get serious, might want to go check that out. Get a karyotype taken to see if they carry that gene. All right, genotype of male, genotype of female. Okay, now we're going to look at some non-Mendelian genetics that take place. We mentioned this a little bit last time about how it was lucky, or providential, if you will, that Mendel chose peas to work on because their traits tend to be handed out on single genes. Had he done snapdragons, he would have crossed a red snapdragon with a white snapdragon, ended up with a pink snapdragon. Because he was kind of thinking that way with the peas, right? That there would be a mixture. Before Mendel's work, that's what they thought. There was a mixture taking place. And it was only through Mendel's work that they figured out there was a dominant gene and a recessive gene on most things. But it turns out what we have right here in snapdragons is something called incomplete dominance. Guys, keep your hands to yourself. Incomplete dominance. And we can see what happens here. We're going to have 25% red, big R, 25% white, little r, 50% pink, big R, little r. Okay, non-Mendelian. He says, huh? But was he wrong? No, but some traits are caused by a combination of genes, by something going differently. So the first one we're going to look at is called polygenetic inheritance. This is how eye color takes place. We know about 2 genes, chromosome pair 15 and 19, interact to determine eye color. On 15, I believe, there are 2 different genes, and on 19, there is 1 gene that all affects eye color. Well, it turns out we've done some very recent studies on eye color, and there's probably up to 50 genes that influence eye color. So eye color is probably not just polygenetic, but possibly one of these other things that I'm going to tell you about here in a minute. Yes?
Since technically color is an infinite spectrum, can we have infinite eye colors, or is it just...
Yeah, pretty much.
So it's not
Because
just a dominant...
it actually has to do with spectrum. And it has to do with the bending of light. Okay, incomplete dominance is when we have created a mixing of traits, such as the color of flowers, red and pink equals red and white equals pink. That would be the snapdragons. That's called incomplete dominance. We have something called epistasis. This is what's happening over here with eye color too, I believe. We have a modifier gene. So we have something being expressed, and then some other gene somewhere will modify the expression of that trait. See how it's getting kind of complicated? Remember I told you genetics was one of the most difficult classes I took in college? That's because all this stuff is coming in to do weird stuff. All right, we also know in some cases a modifier gene will cause no trait to be expressed. For instance, colors in llamas. We have black llamas and brown llamas, and there are genes that affect it that turn out a white llama, and the white llama has a modifier that means no color is expressed, leaving a white llama, which technically is a albino llama. But we see plenty of white llamas out there. Pleiotropy is another one, a single gene. Just the one gene can actually have different, it kind of diverges off and has different traits being expressed through multiple characteristics. So I'm going to take you through these here quickly. The first thing we're going to talk about is polygenes, polygenetic of the eye color. You can see all these different eye colors right there. This is the reason we see so many. So we're going to see something called heterochromia. I told y'all I'd look this up and try to figure it out. This is total heterochromia, is when there's one eye's one color and the other eye's a different color. And you may have known, I know that, who's the rock star I told you? David Bowie had one color, I think he had a blue and a brown eye. And then we have here sectorial or partial heterochromia, where somebody has just part of their eyes blue and part of their eyes brown.
Dogs.
Okay. As an actress who played Dr. Quinn, Medicine Woman, Jane Seymour's her name. She has this one down here. Okay, so here's a guy. There he is.
What the?
Yeah. So what I'm going to tell you is kind of weird. Here's a guy with the partial.
Oh, cool.
That's a better
Right
picture.
there. Yeah, this is a different person. Yeah. So what I'm going to tell you is this is really weird because we think about the color being caused by some sort of chemical that has a color to it. Yes. That is not the case with eye color. There's only one eye color, and it's brown. Weird. Yes.
It's me. So, is that the same as having spots of a different color?
Possibly. That can be considered, like with the thing, like the ring in the middle and stuff like that, probably is heterochromia.
Because I have certain spots, but it's not like it's not like it's a ring of amber. It's like specific spots in my eye.
Yeah, that's probably heterochromia. Okay, so here's what I'm gonna tell you. You might think, well, that's weird. My eyes are blue. How can there only be one color and it be brown? All right, so we're talking about the iris of the eye now. The iris is this colored part. Okay, this round part. That iris, by the way, is also a muscle. It's called a ciliary muscle. It's kind of muscle it's a circular muscle that closes like that, whoop, whoop, like a pull string on a bag.
Is that why it hurts whenever you see
like if you're in the dark and you come out to bright and it hurts? It's because that muscle's getting really small to make your eye pupil smaller to let less light in.
So it's
right now your pupils are kind of big because this room is dark. And if I came and shined a flashlight in your eye, you'd go, whoa, and you'd probably.
Can you video my iris doing that?
So that means it's contracting, right?
It's closing, yeah, to regulate the amount of light that comes in. That's the way our eyes work. Our eyes are allowing a certain amount of light in, and if there's too much light, they'll get if you're in the bright sunshine, you can look at your eyes. They're pinpoint pupil. There's just letting a little bit of light in. Yes.
So then, like, what happens if someone like stares at the sun too long and they go blind?
They go blind. That's the damage being done to the retina, which is the back of the eye.
Yeah. Okay.
Yeah, the light's still getting in. The pupils don't close completely. Okay. Not that there are people that don't have pupils, but let's not worry about those guys. Okay, so here's what happens. In the iris, I want you to look at the iris. The iris, we're looking at just the iris right here. This is that muscle right here.
That's cool.
On the back of the iris right here, which is this part right here, there is a chemical called melanin. Melanin is the stuff that gives you the color in your skin, the color in your hair, and the color in your eyes. If we were to take your eyeball out of your head, where you have blue eyes. If I took your eyeball out of your head and I went to go look at your eye, your eye would be brown. All right, so the melanin layer that's in here, everybody has a brown melanin layer on the back part or the flat end of their iris. Okay, but there's another layer at the front stroma, that's on the top. And on the top, you have varying degrees of melanin. So those of you who have brown eyes have more melanin in the stroma than those of you with blue eyes. All right, if you have really, really, really dark eyes, you have a lot of melanin in the stroma tissue. If you have really light eyes, you have very little melanin in the stroma, and the blue that we see in your eyes is an illusion.
An illusion.
It's the way the light is bouncing through the stroma. It's called the Tyndall effect. If you've ever seen smoke and light shining through smoke, it kind of has a blue tint to it.
Oh.
That's called the Tyndall effect. That's what's happening in our eyes. So really your eyes are just bouncing light off them. There is no color blue in the eye. There is just the appearance of blue based on the light refracting through it, which is
So
bizarre.
it's
Yeah.
not like when you see water on the road in a hot day.
That's not exactly the same thing.
It's a
That's
heat
not
wave.
Tyndall effect, yeah. All right, so do you understand? So that's why we can have these variances of blues. It has to do with how much melanin is in the stroma.
So how come animals other than like dogs and some animals don't have it?
Their eye shape is a little different. Also, animals have weird pupil shapes and stuff, like
What,
hourglass
goats?
pupils and
Like
slits,
goats
like
have
cats have
square
the slits.
pupils?
Yeah, it's really bizarre.
What?
Yeah, goats do.
Like
Yeah.
lizards, but lizards how they like close their eyes like.
Yeah, eyes can be very strange. But this is why eyes are getting very complicated here. It's because of all this. So we think now, y'all, that all the genes to determine how much melanin is in the stromal of your iris, there may be up to 50 genes that are affecting that. I think they know for sure there's like 14 or 15 that they know for sure, but they think there's more. That's why it becomes so complicated. So can two blue-eyed parents have a brown-eyed child? The answer is yes. Typically, they'll have blue-eyed children. Typically. Just because this is still genetic. It's still genetic. Having a lot of melanin in my stroma is still a genetic thing. All right? It's just when they deal the cards out to you, you have less melanin in your stroma, therefore you have lighter eyes.
I have
Yeah.
a question. So my younger brother Jackson, he has like hazel eyes, but like sometimes he'll switch to green. Sometimes.
So that's why this happens. It has to do with the refraction of the light, because I have hazel eyes too. And when I wear blue clothes, my eyes look blue. When I wear green clothes, my eyes look green.
No, like he'll be wearing like something random, but like they'll just change from like green to
And
green.
that's because of the light shining through them.
Yeah, I remember my eyes being more green for like 4 years of my entire
Yeah,
life.
that can happen too. Like little babies can start off blue-eyed and they can actually
But it was
change.
like in the middle
By
of my
making
life.
more melanin, their
Like-
eyes will turn brown
Like
later.
when I
It
was
can happen.
6
I was-
until when I was like 10, I had greener.
And now they're
eyes.
blue?
Yeah, I went from bright light blue to having cyan and hazel.
Oh, really? Okay. Yeah.
Cyanide,
Mine have actually
hazel.
gotten lighter as I've gotten older.
Cyan.
Okay, so we have an idea there what's going on. Let's talk about dogs now. This is another thing called the epistasis. Remember, the epistasis has many influences, like there's a gene. So we're looking at the color of Labrador retrievers. All right, we have a black lab, we have a chocolate lab, and we have a yellow lab. So it says here that we have allele big B for black, and big E is influencing it. So big E comes in and makes the black stick, okay? But a dog that has little b, but big E will turn out chocolate, and the dog who has big B, little b, regardless of its other alleles, will be yellow. So I can't remember I think, isn't chocolate the most rare of the Labradors?
Yes.
Anybody
My
know?
dog's
Yes.
part lab.
I thought it was the
Aren't
most
chocolate
popular.
labs more expensive than
They
black
are
labs?
more
Or-
expensive.
Oh, I thought it was more
Yellow.
popular.
They are, but they're more expensive.
I think they're more expensive.
No, like more, more.
Hey, today was good.
I'm just saying that the reason that this is happening is because it's more than one gene that's affecting it, okay? It's this other one that's called that epistasis. It's influencing that. All right, pleiotropy is one gene coding, guys, for multiple traits. And in this particular case, one of the traits it can code for is a disease called sickle cell disease or sickle cell anemia. And sickle cell anemia is when the cell of the red blood cell is not shaped round like a little raft. It's actually shaped like a moon, like a crescent moon. It's a sickle shape. And if it's a sickle shape, it kind of grabs the vessel as it's going through and catches along. So that's a bad disease, and it tends to show up in people of African descent. It happens to be a gene, that pleiotropy, that takes place to show that. All right, so we're looking, all these things I've been telling you about are the non-Mendelian inheritances, Okay, so we have incomplete dominance. Are y'all supposed to write these down?
Yes.
No.
Result in a phenotype intermediate where traits mix. I wrote red plus white equals pink, but I wrote the word white in black. I don't know why I did that. I think I'd do pink there. Codominance is an inheritance pattern in which both traits are expressed in heterogeneous crosses. Red plus white. Why doesn't it equal pink? We're going to see this specifically. We're going to see codominance is most easily understood when we talk about blood types. Yes.
Okay, so I have a question about eye color.
Uh-huh.
And please don't take this wrong. So, people of the African American ethnicity
Yes.
seem to like. I don't think I've ever seen one with like blue eyes.
Oh, sure, they have blue eyes.
One of my
Yeah,
friends.
Is it like a brown?
One of my
They're
friends
actually very
dominant?
stunning people. Stunning. In fact, if you see somebody who's got really dark hair, so think about this. Have you ever noticed people say blonde hair, blue eyes, right? Because they tend to go together because of melanin. You have less melanin if you have blonde hair, okay? So, but there are people out there who are very dark and have blue eyes, and it's a stunning combination because it's rare. We don't see that much. There are colored eyes that are considered pink in humans. Those are mostly albino people.
Really?
There are people who have colored eyes called violet, which are a purple-looking color. They look like a really deep, beautiful blue is what I say, but they're considered violet. And this kind of thing, yes.
I have dark red hair.
Uh-huh, and blue eyes, right?
We didn't notice.
So how does that work?
The red hair, I didn't do the deep dive on red hair, sorry. I know what happens is typically it will pop out of nowhere. Do you have anybody in your family who has red hair?
Yes. My
My
mom
mom
and
and
my
sister.
dad. And
Well, there you go.
My dad had like a red
And your sister.
and
Yeah.
my grandparents.
Well, so it's very common in your family then.
From what I heard,
Yeah.
Well, not my
red hair
grandparents.
is
my
like
grandparents.
an mutation.
Okay, so let's talk about codominance when it comes to, here's our red and our white making our pink. Okay. Incomplete dominance, we're going to talk about blood types. Do y'all have any idea what your blood type is?
No.
Negative.
I'm not being tested either.
I think that was A.
I think it's O+.
Maybe not.
That's the most common.
Oh, plus.
All right, because here's what happens. If you have type A, you show the tendencies for type A. If you have type B, you show the tendencies for type B. But if A and B are mixed, like a dad with A and a mom with B, instead of one being dominant over the other, they end up with a type called AB. All right, so it's codominance. They are not one is not dominant over another. Although O ends up, the most common blood type on the planet is type O, even though it's recessive. And what happens with O is that it has nothing on its surface in regard to A and B types. Okay, so type A blood will have a spike. This little spike on there is just an antigen, a surface antigen that is anti-B. What they realized on the war front when people had a blood transfusion, sometimes it was miraculous and saved the person's life. Other times it killed them more quickly. And then they finally realized, oh my goodness, there are types of blood. So if you are type A blood and someone gives you a type B blood infusion of somebody else's blood, a transfusion, it'll kill you because you're making a bunch of antibodies against it. So we have surface antigens that make antibodies against B.
So what if it's just like a little bit?
Is there-
Like you have a cut and somebody else's blood spills on it or something?
Yeah, it would probably be very localized and shouldn't be a problem. It's only when you have a full transfusion.
Which one is the universal?
Yeah,
It's like,
it's
you can use
Universal
both.
donor, the universal donor is O because it has nothing on it, so nobody reacts to O being given to it.
If you have AB AB plus blood cancer, you just get donated from literally
Anybody,
anybody.
you're the universal recipient. You have AB.
Universal blood.
What's O negative?
The negative has to do with another factor. It's called the rhesus factor. It was named after rhesus monkeys. RH. It's called the RH factor. Rhesus RH factor, right from rhesus monkeys. And you see here, like for instance, O positive. So I'm O positive, and that means that I can only get O blood and O positive blood at that, you know. But the deal is, if you're O negative, you literally have nothing on there. That's the absolute universal donor right there, O negative. But you cannot get any. You can only get O negative blood. I can only get O positive blood. Actually, I guess I could get O negative blood.
But you can give blood to anyone.
I can give blood to anyone. But I will tell you this, the best thing to do, because my O positive blood, I'm still making antibodies against A and B. So my blood will have some antibodies in it, right? But it doesn't have any antigens in it. That gets a little beyond this course. My Biology 2 course, we study it in great detail. Alright, so you can see what are blood types anyway. Surface antigens are substances that, when introduced into the blood, trigger a production of an antibody, which is a specialized protein that attacks specific antigens of the thing that it made the antibody against. Antigens and antibodies determine blood type. We'll watch a quick video and then it'll be about time for our lab. In my biology 2 class, we actually do a blood typing of my students so they can determine what their blood type is.
Ooh,
It's no fun when I do it because with O, it has no surface antigens, therefore nothing happens when you mix the chemicals together. It just stays the same. So nothing happens. If somebody's AB, everything clots up and it looks super cool. So if anybody knows they're AB, I'd love to have some of your blood. We do it, we poke their finger. Yeah. Some students are not willing to do it, and that's okay.
See, that's how
If we have time
that's how
today-
the vampires
Wait, does it
survive.
require needles?
It's a little finger prick.
Yeah.
Like that.
so.
Yeah.
That's how the vampires
It
survive.
doesn't become viral if
It
you
really,
teach
yeah, it
it.
doesn't hurt really.
Today, if we have time, I have a simulated blood typing we can do. It's not real blood, and it's not your blood. Okay, let's watch a little video on blood types. That was intended.
It's often said that despite humanity's many conflicts, we all bleed the same blood. It's a nice thought, but not quite accurate. In fact, our blood comes in a few different varieties.
Why?
Our red blood cells contain a protein called hemoglobin that binds to oxygen, allowing the cells to transport it throughout the body.
Why?
But they also have another kind of complex protein on the outside of the cell membrane. These proteins, known as antigens, communicate with white blood cells, immune cells that protect against infection. Antigens serve as identifying markers, allowing the immune system to recognize your body's own cells without attacking them as foreign bodies. The two main kinds of antigens, A and B, determine your blood type. But how do we get 4 blood types from only 2 antigens? Well, the antigens are coded for by 3 different alleles, varieties of a particular gene. While the A and B alleles code for A and B antigens, the O allele codes for neither. And because we inherit one copy of each gene from each parent, every individual has 2 alleles determining blood type. When these happen to be different, one overrides the other, depending on their relative dominance. For blood types, the A and B alleles are both dominant, while O is recessive. So A and A gives you type A blood, while B and B gives you type B. If you inherit one of each, the resulting co-dominance will produce both A and B antigens, which is type AB. The O allele is recessive, so either of the others will override it when they're paired, resulting in either type A or type B. But if you happen to inherit 2 O's, instructions will be expressed that make blood cells without the A or the B antigen. Because of these interactions, knowing both parents' blood types lets us predict the relative probability of their children's blood types. Why do blood types matter? For blood transfusions, finding the correct one is a matter of life and death. If someone with type A blood is given type B blood, or vice versa, their antibodies will reject the foreign antigens and attack them, potentially causing the transfused blood to clot. But because people with type AB blood produce both A and B antigens, they don't make antibodies against them, so they will recognize either as safe, making them universal recipients. On the other hand, people with blood type O do not produce either antigen, which makes them universal donors, but will cause their immune system to make antibodies that reject any other blood type.
Is that a bad word?
Unfortunately, matching donors and recipients is a bit more complicated due to additional antigen systems, particularly the Rh factor, named after the rhesus monkeys in which it was first isolated. Rh plus or Rh minus refers to the presence or absence of the D antigen of the Rh blood group system. And in addition to impeding some blood transfusions, it can cause severe complications in pregnancy. If an Rh negative mother is carrying an Rh positive child, her body will produce Rh antibodies that may cross the placenta and attack the fetus, a condition known as hemolytic disease of the newborn.
Is that right?
Some cultures believe blood type to be associated with personality, though this is not supported by science. And though the proportions of different blood types vary between human populations, scientists aren't sure why they evolved, perhaps as protection against blood-borne diseases, or due to random genetic drift. Finally, different species have different sets of antigens. In fact, the 4 main blood types share by us A tree in comparison to the 13 types found in dogs.
Double. And humans donate blood to dogs.
Dogs ain't but a human.
Wait, so is what he said about it attacking the fetus, is that why there's like miscarriages?
Sometimes, yeah. And so I've had at least 2 students, female students, who have discovered they're Rh negative taking my biology 2 class. And that's important to know. By the way, it's not important. It doesn't matter for the first child. It only matters for the second child.
Really?
Because in the first child, the mom is just getting used to it. You know what I'm saying? She starts to make the antibodies then. So it's the second child that's the problem. And so what they do when they know that about the female, knowing that she's Rh negative, they will give her certain medications to tamp down that response in her body toward her child. So a mother can have a child with a different, it's the biggest problem is the mom is Rh negative and the baby's Rh positive.
With a few, so what about the negatives and the positives? Like A negative and A positive?
The negative refers to the Rh factor. Okay, so if I'm O positive, I'm O with the Rh factor. If you're O negative, you're O without the Rh factor. If you're A positive, you're A with the Rh factor. The vast majority of people are Rh positive. Rh negative is fairly rare. Yes.
Is B negative rare?
I think it looked that at the end of the video there, it showed you the most rare is AB, and so AB negative would probably be the most rare. Yeah. So B negative is probably fairly rare. Are you B negative?
No, my mom is.
Is she? Okay. It's good for her to know that. And she had lots of babies, so she probably had to deal with that during her pregnancies. Yeah. Okay, so we can see here what happens is we have a surface antigen that's on the surface of the red blood cell, and then we have the antibodies that are produced in response to it. All right, so we can see here type A will have AA or AO, type B will have BB or BO, type AB will have AB, and type O will be OO. And their phenotype will be AB, AB, and O. And then we can donate to,
Oh shoot, we gotta write this down.
A can donate to A and AB, B and AB, 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 and O.
Yeah, we do.
It says different words.
I don't know if you have to do that.
We gotta write that down.
4 different.
Oh, we don't have to do the phenotype one.
Oh my god.
I'm bringing around to you a take-home quiz.
Whoa.
Wiz.
You need to bring it back for me to give credit to you.
Miss
You may use your card.
Miss
No.
Catherine?
Yes,
Does AB stand for Antonio Brown?
it does not.
Okay. Did you hear there's a warrant out for his arrest?
Yeah.
I did hear that.
For
It's
what?
very sad.
Who's that?
For a charge battery.
Like that.
Who?
What? Assault battery.
There's no clue who that is.
Charge.
It's a football player.
I don't pay attention to football.
That's what it's called, charge battery.
Cowboys had a really good game. All right, and also after you finish writing it down, we're going to do a lab where you're going to taste things. And there's a gene that allows you to taste some things and some things not.
Oregano?
No, it's whatever's implanted on these little papers right here.
Impacted on paper.
But
Please
we're
say
going to
it's
all
not
start
ball.
with the control paper. Yeah. Which is that?
You ever seen that one? This guy does a personal Jesus. So kissy guy.
I'm not quite done here.
Yeah, there's still.
We have lots of notes to go.
Yeah.
Okay, well let's get through this. Can y'all take a picture of it or something?
Yeah, I can.
I'm almost done, please no. I'm almost done writing all of this.
We probably won't have time to do the simulated blood typing.
Yay.
Yeah.
All right, are we good?
No.
Yeah.
Yes.
No.
Yes. Yeah, we're good.
No.
John is wonderful.
Do I have all these words in the same order as your book?
Yes. Except there's no phenotype. There's only
No phenotype? Okay.
Okay.
can donate to everybody. It's kind of a strangely arranged table. Are we good now, everybody? Okay, here we go. The universal donor blood type is O. The Rh factor is another antigen to track the blood types. If a person does not have the Rh antigen on their plasma membrane, they are called Rh negative. Most of the population is Rh positive.
Ooh.
That would be.
Okay, so here we go. The Rh factor is just another surface antigen that is a marker on the cell. And if it's Rh negative, there are none. So O negative is kind of, in respect to types, in respect to blood types, is naked. It's naked on the outside. No surface antigens for AB or Rh factor. All right, so Punnett squares right here. If we have a heterozygous B with O dad and a heterozygous mom with AO the mom, then we're going to see how that comes out in our Punnett square. So we have 25% are AB, 25% are type A, 25% are type B, and 25% are type O. That's why you can have all sorts of different blood types in your family. My husband and I are both O, and all of our kids are O. That's all I had to give them. That's all we had to give them. All right, but I know for a fact that one of my grandchildren is A positive. So that means that her dad, my son-in-law, must be the culprit there, because my daughter only had O to give to their children. 25% of all types. All right, Rh factor, here we go. Plus minus, plus is dominant, so this would be Rh positive here on top, Rh positive. And this is 50-50, Rh positive and Rh negative. If we have a Rh negative parent here and an Rh positive heterozygous there. Good? All right, codominant, so this is just another example. I have a red, this is really kind of dramatic here. Red flower, a white flower, and I have a red and white flower. Not pink, but red and white. Okay, that's a type of codominance here. And we have, it happens in cows as well. When you see a spotted cow, when they go with a white cow and a brown cow, and they end up with spotted cows.
That's cool.
That's a type of codominance that happens there.
Black and white?
I think so. Yeah.
How do they breed Oreo cows where they make it just white in the center?
I don't know. That'd be interesting
Does
to
that
find
have
that
to
out.
do with brown?
This one in the middle is called roan.
Does that have to do with brown and red? Or not brown and black?
That happens with horses.
I don't know. I know it happens with chickens too. A spotted chicken, when you have a black chicken and a white chicken, they'll be spotted chickens.
Like a white
Really?
guy?
Yeah.
I've never seen that.
What about dogs?
Dogs can get kind of crazy, especially if they're not in the same breed. If they're in the same breed, they tend to breed true toward the breed. In other words, unless we have epistasis like those Labradors, right, that have that different thing coming in. But typically, German Shepherd puppies look like German Shepherds.
There's Australian Shepherds and stuff, how they have different colored eyes and like splotchy eyes.
Yeah, so that's going to be that weird thing with the mountain.
Different.
Okay, let's talk about some disorders here. We're looking at some autosomal. That means on the 1 through 22 chromosomes, not on the X-linked or sex-linked chromosomes. For instance, if you're lactose intolerant, I thought it told us what chromosome it took place. You literally don't have a gene that can digest dairy.
So you're missing
Missing
one out
a
of
gene.
23?
Yeah,
Can you
one
take
of a gene somewhere.
Can't
Yeah.
you take a pill though that will break it up for you?
It helps to break down the lactose for you. Yes.
Enzymes.
Sex-linked disorders occur in pair 23, like hemophilia, for instance. That's in the X and the XY. Genetic mutation can cause disorders, like for instance, something called cystic fibrosis. It's a genetic disorder. If you've ever seen The Elephant Man.
Oh yeah.
Who?
Yeah,
That's
John
what he had.
Merrick.
Yeah.
He had cystic fibrosis to a very alarming degree.
Well,
He was very strange looking guy.
Yours
Who is
is
the
skull.
largest
skull
person?
looks like that. I thought it was like weird.
No, that's his
how
actual
his
skull.
looked.
Yeah.
Who is the
Yeah.
largest person in history?
Tallest?
Like the largest, tallest, largest.
Not sure.
What?
Okay, here we
Oh
go.
God.
Damaged chromosomes can cause change in the structure. Listen. It can cause change in the structure of the actual DNA, okay? We have one we'll look at here in a second called cri du chat. Cri du chat means cat's cry. This is a baby when they're born and their cry sounds literally like a cat meowing. It's quite
My
amazing.
brother had that.
The chromosome, sometimes we'll have, that's a chromosome damage that takes to produce the cri du chat. Chromosome number changes can cause disorders. So for instance, Down syndrome is that when there's an extra chromosome number 21. It's called the extra chromosome factor.
Does cri du chat go away?
No.
Well, the reason it's called that and the reason why we recognize it is because when a baby cries, they sound like a cat meowing.
Right.
They have other disorders, so that
Okay.
it never goes away. Do they stop crying like a cat? Yeah, because they stop crying.
Right.
Yeah,
What about if they cry, like if they, I don't know, if they're like 18 and they, I don't
because
know.
we cry
Their
sometimes.
parents
Yeah,
die.
I don't know. I don't know, to be honest with you.
I mean, I could test that out.
What?
Okay, this happens here. Abnormal cellular events in meiosis lead to either none or too many of a particular chromosome. Sometimes there's none. Sometimes there's too many. All right, so we
I
call
thought we
that.
had 23. Why does it say extra 21?
On number 21,
So
on the
there's two
autosome in
21s.
21, there's 2, but this is when there's 3. You end up with Down syndrome. Okay, so for instance, the allele mutation, the actual mutation in the allele. For instance, something called Hutchinson-Gilford progeria syndrome. The chromosome structure is changed. Oh wait, this one's a allele mutation, and we end up with a condition called Hutchinson-Gilford progeria. This is a little boy who aged very quickly.
Are you sure that's
Yeah.
not?
Make it mine.
It's a little boy. I know, but it's very sad because his lifespan, of course, was much shorter.
Is that my father?
So he grows old very, very quickly. That's progeria. The chromosome structural change, the cri du chat, is when there's a deleted region of the chromosome pair number 5. There's a deleted region right there, and it ends up, here's a little girl with cri du chat. Now look at her, she looks pretty normal, right? She cried like a kitty cat when she was a baby, right there. But now I'm sure she lives a life that's just fine. Now if they have an extra chromosome 23, we end up with a condition called Down syndrome,
Right.
which we know, you probably know somebody who's Down syndrome. I have a student who's Down syndrome. She's delightful, so they have certain characteristics that are very, in fact, when I was growing up, they called them mongoloid, which is not a word we use anymore. And that's because somebody from Mongolia who has sort of slanted eyes. So the eye slanting is one of the deals. The tongue too big for their mouth is one of the deals. They tend to not grow very tall, this kind of thing. And of course, it's with them their whole life. All right, so what about gene technology? We can do some DNA profiling. We have something called the PCR, which you've heard a lot about PCR. The polymerase chain reaction is a type of test where you can test genetically, and that's what they used, of course, for the COVID test. We have genetic engineering. We have recombinant DNA. These are all things that are being used to try to, quote unquote, improve our DNA. Sometimes when we mess with it, it's trouble. What causes mutations? Various things can cause mutations, including excessive exposure to radiation. So if you're an X-ray technician, for instance, you're going to be wearing a lead apron instead of being exposed to all sorts of radiation. If some people say, I will tell you there's not really a tie to skin cancer and the sun, although if you have skin cancer, sometimes the sun can make it grow more quickly. Okay.
Sunburn and you.
And they don't have a tie specifically to that.
That's what
I'm not
happened.
saying it doesn't, I'm just saying that they have not found a mechanism.
That's what happened to my great grandma.
What happened?
She had really bad skin cancer, and it got to her lymph nodes and stuff, and all through her body.
Was it melanoma?
Yeah, it was like.
Yeah, melanoma is the worst. But what I'm saying is we don't know for sure if the sun, we can't say the sun caused it, okay? We can't say the sun caused it. We can say the sun may have complicated it, but not caused it.
Maybe it was already in your genes and
Maybe so, right. There are some cancers that we know are definitely genetic. Breast cancer, ovarian cancer are 2 of those. Not every breast cancer or every ovarian cancer, but they can be genetic. Yes?
Can people do a surgery or something that makes them less, that makes their percent chance of having breast cancer less?
If they have their breast removed. If the breast tissue's gone, it doesn't have cancer in it. That's a very dramatic choice. Some people have chosen that. I'm not gonna blame. Yes?
It's gonna sound stupid, but how is smoking and lung cancer directly related?
It's because the cancer is the overgrowth of cells, and so if you think of it kind of like an oyster making a pearl, the reason the oyster makes a pearl is because it's irritated, and it has a little piece of sand in it, and it starts to build the tissue around it. The same thing happens in your lungs. If you have a lot of particulate matter going into your lungs, our lungs start to build this tissue around it, and when the tissue's building around it like that, it says, oh, it's time to grow, and it starts to grow, and it turns sometimes into cancer. Not everybody who smokes get cancer. I will say smoking is very bad choice, very bad choice, don't make it.
Smoking and
So, what percentage of people?
I don't know the answer to that, I'm not sure. All right, here we can see here using gel electrophoresis. If you've ever watched a crime show, you may have seen this. We can take somebody's DNA, put it through here, and it kind of settles out by weight. That's an easy way of saying it. It settles out by its weight. Fragments create a pattern that looks something like this. You can match that to a crime scene and figure out if somebody might be a suspect in a crime. The PCR stands with a primer, and the DNA is heated and cooled to open a strand, and we take these strands and make copies in order to follow something. We call that the polymerase chain reaction, and the PCR would be the polymerase chain. In the COVID test, the R was something else, because it wasn't the reaction so much as it was something else. Okay. Recombinant DNA is when bacteria can be used to actually introduce a new gene into something. This is where we're getting into the area of science that you might call Frankenstein, where we're making different genes out of things. Typically we don't do these on humans because that would be unethical, but we do especially in the plant world, trying to make corn, bigger, taller, stronger, grows faster kind of a thing. We might use recombinant DNA to do that. The question is, has it changed it enough? Sometimes it changes enough that my body doesn't recognize it. I believe that is the case with wheat, that our body doesn't recognize wheat anymore, and that's why we have so many people with wheat allergies or gluten allergies. So, I'm going to say messing with the genes of something is probably a bad choice. Okay, it's lab time. Did you have a question, David?
So, like